Hi, my name is Suresh Pokharel. I am a PhD student at Michigan Technological University. Today I am going to present on predicting protein succinylation sites using a language model-based features. This is the outline of the presentation that I am going to present today. Let's begin with the introduction of post-translational modification. So post-translational modification is a phenomenon where amino acid sites and modification occurs in protein after translation process or their biosynthesis. There exist almost 400 different types of post-translational modification, and succinylation is one of the important post-translational modification that occurs when succinyl group is added to a lysine residue of a protein molecule, and that post-translational modification is called succinylation, PTM. Now let's talk why it is important to predict succinylation in protein. One of the main reasons is it is responsible for vital metabolic activities in cell like cellular respiration, regulation, and repair. Next is the presence of succinylation may lead to the dysfunction of vital biological processes in our body. Talking about existing tools, most of them has used handicrafted features like amino acid composition, and some of them has used secondary structure informations. Also, some of them have used embedding layers of Keras. In this work, our contribution is we have used a pretend language model called 45XL Uniref along with different deep learning architecture to predict the presence of succinylated site in the protein. Talking about the dataset that we used in this research, all the sequences were curated from UniProt-KB, and we used CD hit with cutoff similarity 30% to get rid of the homologous sequences, and 5,009 succinylated sites were extracted from 2,322 different protein sequences, and they were further divided into training set of 4,750 and test set of 254 succinylated sites. And once the positive sites were fixed, then negative sets were extracted from the same proteins that have lysine K residue that is not listed in positive training and testing sets. So this figure shows the number of succinylated sites and non-succinylated sites that we used to train our model. So the basic idea in this work is we have protein sequences, and we feed that protein sequence to the 45 pretend model, and we get a feature vector of size 1024 from that model. And furthermore, we use that features to train the different machine learning and deep learning architectures. As you see in the figure, we extracted 45 features and we trained random forest, support vector machine, and XGBoost, 1D CNN, and artificial neural network. Then we compare the results of all these architectures. Let's briefly discuss what 45 features is. So 45XL Unit 50, this is the name of the model, and it is trained on Unit 50 consisting of 45 million protein sequences. So it is developed by Roche Lab, and the model is based on Google's transformer model called T53B that has 24 layer encoding decoder, and it has almost 3 billion learnable parameters. As we already seen in the picture, model takes the sequence as input and returns an embedding vector of dimension 1024 for each amino acid. Therefore, for each succinylated site, we have 1024 features. Let's move to the result section. Here you can see the tenfold cross validation results of different 5 models that we trained, and we use accuracy, MCC, sensitivity, and specificity as the performance metrics to compare these models, and we found that artificial neural network has the best performance uh, here. Also, we plot the receiver operating curve, and we can see the artificial neural network has the best area under the curve as compared to others. So coming from the tenfold cross validation of previous slide, we found that this configuration of artificial neural network is performing good as compared to other models we tested. So this model is our final configuration for predicting succinylated sites using 45 features. Now to determine how our model performs on unseen data, we used a balanced test set of 254 positive and 254 negative sites to evaluate our model. Also, we compared the results with embedding layer based model called deep succinyl site. As you can see in the table, our model has shown the competitive results as compared to deep succinyl site. So the conclusion of this work is 45 based ANN model has shown competitive results as compared to existing handcrafted and other models. So we can see the future of language models in post-translational modification prediction works. These are some references, and this ends my presentation. Thank you very much.